Thanks very much, Perry. So that's right. Today I'll be talking about some relatives of the elephants. I'll be mostly talking about mammoths as part of a little mini update on the British Columbia Megafauna Project, which is the brainchild of my supervisor, Dr. Mike Richards, which recently also gave an update to the archaeologist community where he was depicted with a little mammoth by an artist who was watching his talk. And so this project was very much in its infancy at the last symposium in Courtney. And it was so wonderful to have a chance to have a poster there and talk to all the attendees who gave me so much hope that this project was feasible and worthwhile. So a big thank you to everyone from the last symposium and also to you listening now. Okay, so the project is basically kind of tackling this problem or this view of British Columbia. You know, how many megafauna remains in the last, say, 10,000 to 50,000 years ago are around? Did they survive extensive glaciation? And do they still exist? Or are they all gone and ground to a pulp? And again, we're in the archaeology department, so this is new territory for us. And so we were kind of wanting to know what kind of mammals are out there. And these are kind of the maps that we had in our heads of widespread glaciation in British Columbia and not really space for animals to move around. And this is also represented in a lot of maps where you see extinct megafauna like mammoths or bear or bison or elk. And there's lots of records south of the border to us, and then east in Alberta and up north in Alaska and Yukon. And then British Columbia is kind of in this sparse area. So the goal of the project is to see if the sparseness has to do with the fact that animals weren't there, or maybe their fossils or soft bones did get eroded away. And so we wanted to look at where are they? Are they mostly in private collectors' homes or at institutions? And if we can find them and they are where they say they are we allowed to destructively sample them to learn about when they died and also learn about their diet and environment through carbon, nitrogen, and sulfur isotopes, as well as very, very fine grained diet stuff. So I wanted to put this first because this project would be dead in the water without this great team, as well as an incredible host of museums. And I think this represents more than just institutions where this Pleistocene megafauna remains are housed, but it also represents institutions where curators are passionate about natural history and they're willing to have people they don't even know come and destructively sample specimens that may be the prized item in their collection. So absolutely cannot do this without all of these people. So we knew at least that we had something to go on. So this is, these are the extent of the published radiocarbon dates for mammoths in British Columbia. And so we at least knew there was hope, especially in this cluster down south, where we are in Vancouver, and then south in Victoria, a nice cluster of dated mammoths. So we started there, not only because the RBCM, of course, is the largest repository of this material, but also because the Grant Kitty and Victoria Arbor were so gracious and hosted us for a week and longer while they helped us look at their collections. And also there's amazing internal document that Grant produced about more locality information on these mammoths, and also includes tidbits about correspondence from the public to the curator. And here's one talking about oh, there's a mammoth tusk in Courtney and it's hanging in a hall. And when I read this, it wasn't one of the stories I had heard about. And so luckily the Courtney archives, a lot of digitized photos. And so went on there and saw this amazing photo with, okay, here's the hall that was referenced in the letter. And then to the left, you can see this nice curved object, which kind of looks tusk shape. So it gives me a lot of hope. So immediately I contacted Pat Trask, an archivist, who Pat hadn't heard of it. So I was kind of like, oh, that probably isn't what it is. And archivist found a close-up video or photo, which looks promising. And then you look at one of these ends, which kind of flared, which is not really what you want in a tusk, more rib-shaped. And so sure enough, Pat was able to go through the collections at the Courtney Museum and find this. And here it is. This is exactly what was hanging on the wall, and the bolts are still in it. And this story has been repeated over the last 4 years on this project, with many stories exactly like this. You're running down leads and you're running down stories, and then you come to this conclusion that maybe it's not mammoth, which is kind of an interesting, I think, human fossil bias to the record, where not necessarily the animal is what they think it is. It's the adjective describing the size of it. It's mammoth in size. And so we encountered this a lot. And I know Mike Wilson talked about this too at the last symposium about finding a whale where you think there might be mammoth. So how do we deal with this? Some remains that we encountered during this project were really fragmented, and were thought to be mammoth, but we didn't really want to have probable maybe mammoth. We want to know for sure. So we're employing a technique called zooarchaeology by mass spectrometry, which takes, often you use in archaeology, taking unknown artifacts made of bone or unknown bone, and looks at the protein peptide signatures of it, and compares it against a database of known signatures. So you can see if your bone matches up to a known specimen for species identification. And so we did this a few times and found that 5 mammoth specimens actually turned out to be whale, but you can also have it the other way around, where you can get whale, or you get mammoth where you thought it was whale. So it's a fun technique to use when you want to be absolutely sure. But a big part of our project is stable isotope analysis. And so you are what you eat, and we really want to get into the nitty-gritty of what these megafauna animals are eating. So we're looking at the carbon and nitrogen as a direct measurement of their diet. And to the left, you can see nitrogen values on the left plotted against carbon values. And often herbivores, you can see a really low diet of protein. So they're low on the left there. And then as you move up on the trophic level, you can see carnivores are much higher on the nitrogen value. They're eating the herbivores, so they're higher trophically. But mammoths have always been this kind of weird outlier. And they kind of have this high nitrogen value, even though they're grass grazers and they're eating plant material. So not high in protein. So we want to look at this to see how the British Columbian mammoths compare to these mammoths in Siberia and Yukon that have these elevated nitrogen levels, to see if there's something interesting going on, or if it's latitude based, or if it has to do with the baseline environmental data of the isotopes, nitrogen isotopes in the grasses. And so we're also want to look at sulfur isotopes to look at their range. So a few different things to look at diet. And then another PhD student on this project, Kara Kubiak, she's looking at really, really nitty gritty stable isotopes. She's looking at the amino acids to get at, get rid of the baseline environmental data and just look exactly what they're eating. So in order to see if there's enough samples to do all these things, we went on a few trips. One of them started in Vancouver Island to the RBC again. And so Kara's here, you can see her sampling an extinct bear. And here's Grant Kitty holding up a mammoth humerus. And I traveled up island, kind of creeping along gravel pits. And I think this project involves like a lot of creeping and a lot of optimism. And we came to Courtney Museum, where Lauren Pat hosted us. And this mammoth molar you see is found by the lady who wrote that letter about the tusk hanging on the wall. Also traveled to the Okanagan, where previously no radiocarbon dated mammoths have been reported, including going to this amazing gravel pit that's been owned by the same family for over 100 years. They had tons and tons of animals come out of this, and they have a lot of their specimens in local museums, including this beautiful mammoth molar at the Enderby Museum. We also visited the Okanagan Science Center, the Vernon Museum, the Penticton Museum, Kelowna, again, looking for more than mammoths, whatever Pleistocene material they had, really. Just wanted to get as much samples as possible. The Princeton Museum, maybe some of you going on the field trip will see that. Also went up north, lots of material in the Peace River Valley, of course. So here, Fort St. John, another piece of mammoth tooth. BC Hydro was also willing to let us sample a piece of Marty. Maybe some of you have seen this at the Visitors Information Center at the Peace Canyon. And then also, Andrew let us come into amazing collections at the Tumbler Ridge Foundation. And so we sampled, or he sampled actually some material and sent it down after I left too. Some bison and horse and mammoth and some elk. And also Dawson Creek has an amazing display, an amazing Pleistocene material from Alberta and Yukon in here. Also went to Prince George. And it was always amazing because often when I went to these museums, the curators had planned for the media to be there. And so here is Chad with the 6 o'clock news. So it was really nice to be part of kind of a happy news story. Also, very grateful to individuals and institutions who mailed us material during the year of COVID when we weren't able to travel. So we also got samples that way. So now that we have the samples, we have a few of them, what do we do with them? So this is the chemistry lab in the archaeology department at SFU. Oh, and I should say that sometimes you see little museum icons in the bottom, and that refers to if there's samples in the photo, that's the museum they relate to. So here we have an amazing rib sent from Bulkley Valley Museum. And the first step is really to make sure we remove any surface contaminants before we begin the collagen extraction process. So we drill about a gram worth of material, which if it's well preserved like this one is enough to do the zooms, collagen removal, single compound isotope analysis, and DNA. And then the next step, which is my most enjoyable step, is putting in a weak acid and watching the bubbles form and it dissolve. And this is a really important part of the process. You really have to babysit the samples because some labs have a protocol where they remove the samples after a set amount of time, like 48 hours. But it's really not until the tusk or bone kind of bends around your pipette that it's ready to come out. And so the average was about 4 days, but the Courtney molar we saw earlier took 2 weeks. So you never know how long the stuff takes to demineralize and be soft enough for collagen extraction. So next we heat it up, ultra filter it to make sure we're really capturing the material that is the collagen size that we need. And then this is kind of like the do or die moment where you know if there's any intact material inside the bone. You take the frozen samples and then freeze dry it for a couple of days. And then you get this beautiful foamy, sparkly material, which is in our bones. Or to the right of the photo, you get no collagen, which means we can't do any of the isotopes or dating. So it's kind of a nerve-wracking situation there. And then next, we weigh the collagen to figure out what the yield is. We know how much to send away for radiocarbon dating, and then take little tiny capsules that get combusted in this mass spectrometer, where the carbon, nitrogen, and sulfur gases are red, so we can make sure that all the ratios are okay, and there's no contamination, that we know when we send away the sample for radiocarbon date, it's not contaminated and it's going to be a good date. So we do this in-house to avoid extra costs. So it's nice to do it at school. So again, this is the map we started with before we started the project. And then here we go. There's quite a few dates which we saw those empty tubes with no collagen. Unfortunately, it's just a preservation issue. These things, they've been out of the ground for 100 years and they degrade very quickly. And then we have this many dates that are new that we can add to what is known in British Columbia. And I should also say we also have dates for other animals like horses and bison, but these are just the mammoths. And then currently we are still maybe, I don't know, 6 months away or so from a bunch of more dates, mostly up north. And then you can see some more on Vancouver Island, including Victoria. And Victoria is interesting because not only are there a lot, but it seems to have the most range. So one of the oldest ones we have is kind of the limits of radiocarbon dating, about 46,000 years ago, minimally. and then a very young date that Grant Katie got on that humerus that you saw earlier. And so that was an older style of dating. So we're redating it with AMS. So we'll see if it's as young as we hope. Another thing moving forward with this project is hoping to incorporate DNA, because a big question right now is, what mammoth is it? What do you have? And you can see British Columbia here. We have a red dot, and that's indicative of a Columbian mammoth. So the larger, bigger, more southern variety. So what's it doing here? And how do we know what it is? Traditionally, we use limb lengths to extrapolate height, which then puts you into a size category of which mammoth. And then also we use dental metrics, looking at the way the enamel plates measure to see species. But again, as you can imagine, that kind of stuff is obviously impacted by taphonomic factors, and then also, of course, by the dental wear itself. So this is kind of like Instagram view of mammoths. This is the reality of British Columbian stuff. Because we're not, stuff isn't eroding out of the permafrost like up north, it's not as well preserved. So, the post-cranial remains we have to the left, you can't extrapolate any kind of limb or height measurement from that. And then this tooth is a pretty standard tooth that we've encountered, where it's very delaminated and the enamel isn't quite helpful. So we really want to get at this with DNA. And a really interesting place that I hope we can do this is up by Smithers. There was a mining operation that uncovered dozens and dozens of mammoths and other megafauna, that died about 30,000 years ago. And that's where that red dot was from the other map. So the Columbian mammoth was here. And at the time of this discovery, Harrington wrote, "Future mining may expose more fossils." But I think what would be more accurate is future divorces may expose more fossils, because a lot of miners took things home when they were found into, lunch boxes, and then after, say, a divorce, or the wives didn't want it hanging around on the mantle, so they donated it. And one of them has dental characteristics of woolly mammoth. So what's going on here? Do we really have both species? Is there hybridization happening? And then if we do DNA on this, we can get at the sex. So is this a male-dominated, sticky pond sediment trap where they're getting stuck? And because often males are straying from the matriarchal pack and they're getting engaging in riskier behavior. So maybe that's why we have such tall individuals, because it's a male biased assemblage. So yeah.